exactly how your eye takes light, photons, and turns it into something your brain can understand. The brain receives and sends messages via neural pathways. A neural pathway connects one part of the nervous system to another via axons, the long fibers of neurons, basically. An example of a neural pathway is the optic nerve. The optic nerve connects the eyes to the back of the brain, aka the visual cortex. The neurons and axons of these neural pathways communicate with electrical signals. Now, photons are not electrical signals, yet we can still perceive light. So how can that work? The answer? Through a process called visual phototransduction. Visual phototransduction is the process in where the rod, cone, and supersensitive ganglion cells of the eye convert light, the photons, into electrical signals to send to the brain. Rods are cells in the retina of the eye that function in less intense light and perceive mostly in black and white. Cones are cells in the eye that function better in intense light and perceive mostly in color. Ganglion cells are cells in the eye that are like neurons that receive visual information from the bipolar cells and retinal thyroid cells. This process of visual phototransduction is very important because you would not be able to see this video right now. The process goes like so. The incident photon, represented here by HV, is absorbed and activates a rhodopsin, a purplish red light sensitive pigment, by a conformational change in the disc membrane to activated rhodopsin. Next, the activated rhodopsin makes repeated contacts with transduction molecules, catalyzing its activation to the G protein by release of bound GDP, guanosine 5-diphosphate, in exchange for cytoplasmic GTP, guanosine triphosphate, the A and Y subunits. Then, the G protein binds inhibitory Y subunits of the PDE, phosphodiesterase, activating its A and B subunits. Next, the activated PDE hydrolyzes, which is the chemical breakdown of a compound due to reaction with water. So it hydrolyzes cGMP, cyclic guanosine monophosphate. Finally, guanidinol cyclase, GC, synthesizes cGMP, the second messenger in the phototransduction cascade. Reduced levels of cytosolic cGMP cause cyclic nucleotide gated channels to close, preventing further influx of sodium hydrochlorides and calcium cations. Without this process, during times of distress, we would not be able to see the light at the end of the tunnel, or be able to tell if it was an oncoming train. In addition to the discovery of this process, it has shed light on retinal diseases that can or may not be hereditary. The G protein has been linked to many eye conditions, such as gene mutations, color blindness, retinitis pigmentosa, and congenital night blindness. Scientists are now studying this protein to cure these diseases. Can you see the light?